Okay, so happy Tuesday, everyone. Welcome to another installment of Paleo Perks seminar. And today we are joined by Bethany Allen from GFZ Helmholtz Center of for Geosciences from Germany. And today she is going to talk to us about how can we quantify biodiversity in deep time. For those of you who are new or joining a seminar after a long time, we will go over the format of today's seminar, which welcome and announcements, which is happening right now. Then we'll have the talk for 30 minutes, followed by a moderated question and answer session. Don't forget to send your questions via chat to the questions at PaleoPerks host, who today is Nina. And then our speaker has agreed to stay with us for the informal tea time. Early career researchers are especially encouraged to join during this informal chat. A bit of housekeeping before we dive into today's seminar. Paleo Perks values the participation of all folks interested in the paleo sciences. Please remember to abide by our code of conduct during today's seminar. You shouldn't be able to unmute yourself, but in case you do, please mute yourself for the duration of the talk out of respect to our speaker. You can ask questions by chatting to the questions at PaleoPerks host or by using the raise hand function. If you face any technical issues, please do not hesitate to reach out to the questions at PaleoPerks host. We have closed captions built into Zoom. Use the CC button to show or hide them. Paleo Perks and promotes the work of early career researchers. And so please consider nominating an outstanding ECR in any paleo field. The link to the form will be dropped shortly. We also collect anonymous weekly feedback to understand the demographic of our audience and other interests. The link to this feedback form will also be dropped into the chat window shortly. To introduce today's speaker, Bethany has done her bachelor's in science in biology and earth sciences from Durham University in UK. Then did her master's in science in paleobiology from University of Bristol, UK, followed by a PhD at the University of Leeds in UK. Then she did a postdoc at ETH Zurich in Switzerland, and currently she is a postdoc at GFZ Helmholtz Center for Geosciences in Germany. Bethany, the floor is yours.
Thank you very much. I will just share my screen now. And now that should be in full screen. Is that correct?
Yes.
Brilliant. Okay, in that case, I guess I will take it away. So thank you so much to PaleoPerks for inviting me to give this talk. And thank you to all of you for joining me today. So I'm going to be talking to you about a project that I've been working on with Rachel Warnock and Alex Dunhill, two of my longtime collaborators and people I think a lot of. And we set out to kind of survey the landscape of previous research on how we can quantify biodiversity in deep time. And so what I'm going to present today is essentially a summary of a review paper that we currently have in review, but the preprint is also on Evo Archive. You can follow this QR code, which I'm going to put up at the end of the talk as well. If any of this has interested you or you want to read up in a bit more details, we would love for you to take a look, and we'd also really love for you to send us any feedback. So if you have any kind of thoughts following on from this talk, then please do get in contact with me, and I'd love to hear from you. Okay, so in terms of an overview of this talk, so I'm going to start out by giving you a little bit of background that I think probably most people in the audience already know on why reconstructing ancient diversity is difficult. Then I'm going to go over some of the methods that are presented in the review paper in terms of how we can estimate diversity using the fossil record. Then I'm going to go through the example that we present where we test out using these different methods on the fossil record of brachiopods in the Permian Triassic, and we can see what these different methods do in terms of recreating a diversity curve. And then finally, I'm going to go over some recommendations that we drew from kind of reviewing all of this literature in terms of how we can move forward as a field and hopefully come up with even better metrics and approaches in the future. Okay, so let's start off. Why is it hard to reconstruct diversity in the past? So I have used this figure in many of my talks. I really, really like it. But essentially, if we think about all of the life that is lived in the past, a tiny, tiny fraction of that is going to end up in the fossil record. So if we look at this figure, we can look at the gray rectangle representing the reality, this kind of all of these many, many animals and plants that have lived in the past that describe this true diversity that we might be interested in learning about. But then the actual information that we have in the fossil record is represented in this white rectangle in the bottom left. So we have this dashed record, which represents the known fossil record. So this is kind of all of the information from fossils that we already have. You might think of, for example, all of the fossil data in the paleobiology database, for example. And this represents kind of our complete-ish data set of what we know, what we think we know about past diversity. And then as we go on to collect more fossils, collect more data, we might conceivably reach a point where, uh, in the infinite future, I suppose, reach a point where we have collected all the fossils there are to collect. Um, and and perhaps that inference might change compared to the results that we might get if we just looked at the fossil record that we have now. But then how does that also map onto this bigger gray rectangle of reality, all of these animals and plants that have never reached the fossil record? And so I guess a lot of this work that I'm going to present to you today, these are methods that people have developed to try and go from this dash rectangle in the bottom left and infer things instead about this gray triangle. And the methods are about bridging this gap between the two. And so there are many, many ways in which the fossil record is incomplete and biased. And these are the things that we need to overcome in order to get this reliable signal for biodiversity through time. So on the left, this is summarized using a figure from this paper by Wall et al. That really is a summary of how much rock we have of different ages through time, and how this serves as a filter on how many fossils we can collect from different ages. So, for example, here you can see we have a real peak in the cumulative area of rock about 100 million years ago, and this means that we have much more rock area to look for fossils in for the Cretaceous, for example, than we might do for the Cambrian. And so this means that if we want to compare diversity between the Cambrian and the Cretaceous, then this amount of rock area that we have is one of the ways in which we're not making a fair comparison there. And another way is that we do not have even sampling by geography. This is exemplified on the right, showing a map of all of the points where we have fossil data in the paleobiology database. And you can see that for the US, for example, we have bigger circles showing more fossils and more points showing more localities in comparison with somewhere like Africa or South America, for example, where we have kind of less contribution to our global fossil record in terms of collected fossils and also those fossils entering our data sets. So that's a little bit about the biases. So what does this data actually look like? So typically when we're thinking about the fossil record, I think most people would consider it to be something like this schematic that I have on the screen here, where we're thinking about fossil occurrences. We're thinking about specific fossils that have an identity in terms of the taxon that they belong to. And we might have only one of these, or we might have several of these that correspond to this taxon persisting through time. And this is kind of the raw data that we might be using in order to infer that biodiversity. But then another way that we can think about the data that we might use for thinking about biodiversity in deep time is also through phylogenies. And so these phylogenies might be built only using these fossil occurrences, especially if we're interested in extinct taxa. But then if we're thinking about something which is more recent, then genetic sequences, for example, can also provide information on the relationships between these taxa. And this therefore feeds into our understanding of time and diversity through time as well. And so these are kind of two different ways that we can use data and bring data to this question that can, in some ways, be complementary. And I guess the ways that these two different sources of data can be used is elaborate a little bit more within this review paper. So now to talk about the methods. So in our paper, there is actually one big, like huge table that describes all of the different methods that we have thought about or considered in the literature, which are used for estimating diversity in deep time. So there are 24 different methods or metrics. 10 of these estimate paleodiversity. So these are methods that try to answer the question, how many species were there? They're looking for a number of species or genera, for example. But then there are also 16 methods that estimate rates. So this might be origination, speciation, extinction, diversification, these sorts of things. So these are the kind of methods that we might use if instead our question was something like how quickly was the number of species changing? Or we might use modifications of these approaches to ask questions like what proportion of species went extinct at a specific event or time interval. And so these are kind of two different but complementary ways to ask questions about diversity and diversity change through time. Okay, so that's a kind of summary and overview. And so now I'm going to go into a little bit of detail about how there have been kind of different schools of thought on how to address this question and what these are. So, kind of the simplest thing that we can do is an approach called ranging through. So, if we think about our raw fossil occurrences, you can imagine a scenario where we have a taxon that exists in time interval A. We then have a following time interval B where we don't have any fossils of that taxon, and then a time interval after that, C, where actually we do have fossils. And so, under our kind of traditional species concept, we would assume that it is not the case that this species kind of arose, died out, and then arose again. We would instead assume that actually it still did kind of persist through this interval. And so instead, we're going to say it doesn't exist in B because we haven't sampled it. It's because the fossil record is incomplete. And so we would fill that in by ranging through this clade. And so we might refer to this interval as a ghost range, or we might refer to the taxon as a Lazarus taxon, where we have this interval where we don't have it actually sampled, but we would consider for the purposes of our metrics that it was there. And so we can add that kind of extra one in our tally for that interval. So just to kind of briefly mention, usually when we're thinking about ranging through, we're considering using fossil occurrences and where we can directly fill in those gaps. But it's also we're thinking about the fact that also phylogenies can be incredibly useful for filling in or at least creating a model where we can see points in time where we think that a species or a genus did exist, but actually we don't have fossils. So you can see in this example on the right-hand side where we might have a fossil for our species in time interval C, but then if we infer a phylogenetic relationship with that species with another one from which it diverged X amount of time ago, then we can also use the length of that branch in order to infer something about when that species existed and therefore kind of go beyond these gaps that we might have in the fossil record. And so ranging through is something that's been done for a very long time. In this kind of classic example of this diversity curve from Raup and Sepkoski from 1982, this is the kind of approach that they used. They collected all these different occurrences of their marine families through time and then filled in some of those gaps based on kind of knowing that it was just the thinness of the fossil record and not that things were kind of dying out and then reappearing again. So this is kind of a really good foundation for some of the methods, the more advanced methods that we have seen in the literature that have been developed for this question. And so one of these is the kind of family of methods known as cohort analyses. So we've already introduced this kind of idea on the left-hand side where we have these kind of intervals of time where we know that our taxon does or does not exist. And so we can take that forward and again view our fossil record as a series of time intervals, maybe geological intervals is a very traditional way that our data kind of exists. And then for each of those intervals, we can say, is our taxon there or is it not? And so we can use different ways to kind of codify this. So in this example in the middle, this is based on kind of foot rates, if you will. We might have our time interval I, where we might say this is the only time interval where this species exists, and then we can identify it as a singleton. And then we might have different cases, for example, where we say, oh, here we have a species that where the first occurrence corresponds to this bin, but then it continues on afterwards. We might have other scenarios where we have older occurrences, and this is the last bin in which we have an occurrence of this taxon. And so we can count up all of these, all of the species for which we have these different scenarios, and then use these to try and account for some of these gaps and correct the fossil record somewhat, if you will. And so a lot of the methods that have been developed by John Alroy lean on this thinking as well, the kind of three timer and second for third, these kind of approaches. These also use a cohort analysis kind of style of thinking where each of our occurrences is placed into a bin. Um, the range of that taxon may take a different shape or form, and then we count up the different shape or forms that we have and use this to make some sort of correction on the incompleteness of the fossil record. So just to kind of briefly mention, traditionally this is something that is done on a global level and is used to estimate things like changes in diversity across mass extinctions, for example. I also did some work during my PhD where I kind of investigated whether there's any scope for kind of applying these metrics for looking at differences in diversity across space. This is a kind of schematic which describes the kind of moderately complex way in which you have to do that. Essentially, it's very difficult because it's really hard to differentiate between when a taxon is missing because of the paucity of the fossil record versus because it migrated somewhere else or because it went extinct. And so, this approach very much definitely has its pros and cons. So now I kind of want to come back around to this idea of fair sampling that I introduced earlier. So we might imagine a scenario where we have been out and collected some fossils, and let's say that we have a certain number of fossils for the Triassic, a different number for the Jurassic, slightly higher, say, and then a certain other number for the Cretaceous. And we might have different species diversities that correspond to each of these number of fossils. But then we might look at this record and say, but wouldn't it be fairer to be comparing the same number of fossils between our different time bins? Because then, if we had the same number of fossils but a different amount of diversity, surely that would give us more or a fairer insight into differences in diversity through time. And so there are kind of two approaches that we can take to this. So one is known as rarefaction or subsampling, where we would kind of take for our smallest bin, so in this case the Triassic, we have the fewest number of fossils, and say however many that number of fossils is, I'm only going to look at that number of fossils for my other two bins. And so for any kind of additional ones above that, we would just discard that data. And then in that way, we're comparing diversity between the same number of fossils across our time bins. We can also do the opposite. So in this example, we could say, well, for the Jurassic, I have lots of fossils. I don't want to throw them out. I don't want to discard them. But I also want to make a kind of fair comparison between these bins. And then in this case, we can use approaches which extrapolate our data. So we look at what we already have and we make inferences about what we would see if we instead had this higher number of fossils, and do that in order to make some sort of fair comparison. And a lot of the methods actually that are really popular at the moment use either subsampling and or extrapolation. So some of these in particular are coverage based. So if you've heard of SQS, for example, this is a type of coverage based method. So to kind of I'm going to briefly go through an example where I explain what it means for a method to be coverage based. So we can imagine a scenario where we have found a quarry that nobody else has found before, and we want to take a series of field seasons to collect fossils from this quarry. So we can imagine that we might have this graph where we have time or effort on the x-axis, and then the total number of species that we've discovered from this quarry on the y-axis. So you might imagine that as the number of times that we visit this quarry increases, the number of species that we're going to have recovered from this quarry increases. But what is the actual kind of shape of this relationship? So we might expect that early in time, kind of the initial fossils that we're finding, everything that we're finding is new because the kind of the backlog of what we already have doesn't exist. You know, everything we're pulling out of the ground is fresh. But then maybe we've been to the quarry 10 times, 20 times, and then it's more likely the case that each new fossil that you pull out of the ground actually is more similar to something that you already have. And so you're not actually adding to your species diversity as you find these fossils. And if this is the case, then you might end up with a relationship that kind of looks like this, where you might approach some sort of asymptote. So you would expect that actually, once you kind of reach this flat line at the top, this might be a fair description of the total species diversity that you could ever possibly hope to get out of this quarry. And if you just keep going back, you're just going to keep on finding the same old things. And this is kind of summarized using these metrics. So you can see on the left-hand side, if you kind of picked up 10 fossils at this point in the graph, you might expect to see 10 new species, whereas on the right-hand side, you might pick up 10 fossils and expect actually to find no new species. Everything is something that you've seen before. And so we can imagine a scenario where we're somewhere in between these and we want to have an understanding of how well we have sampled our quarry. We can imagine a scenario where we have found 10 fossils and actually within those 10 fossils, we have 4 new species. We can use this information to kind of put ourselves somewhere on this line. And also potentially we can use the information that we have gained along this line up to this point, so kind of the left-hand side of the graph, to take an estimate of where that asymptote might be or how far away it is, how many more times we might have to go to this quarry, and then, yeah, some sort of estimate of the total diversity that we might have. And so this is the idea behind coverage-based methods. So essentially what we're doing is we're using the shape of this curve where we're kind of sampling more fossils and seeing how much diversity is contained within those when we add those to our total number, and then using this to make an estimate about how well sampled our locality is. And then we can kind of, if we can identify this same line between our different quarries, then we can use this to make some sort of fair comparison. And so in SQS, we might call this a quorum level, where we're saying, well, I want to compare the diversity that I have between this dashed line on each of my quarries. And then when I'm doing that, I'm comparing the same amount of sampling effort. And that might be considered fair. And so, as I say, this is an approach that has been used quite widely in the paleontological literature. Here's a really nice example led by Emma Dunn, where you can see on the left-hand side, we have our kind of collectors curves. So they look a little bit like this kind of schematic curve that I just showed you before. So this is telling us something about if when we kind of sample the fossils that we have, for each new fossil that we sample, are we adding a new species or not? And then when we characterize this curve, it can tell us whether we are approaching that asymptote or not. And then we can use that information to infer our total richness on the right-hand side, or at least a level of richness that is comparable between our curves. So in this case, you can see for the kind of Carboniferous to Permian boundary, where we have these different curves with our different quorum levels, those dashed lines, giving us comparable estimates of diversity through time. Okay, so to follow on from this, there was also a kind of line of thinking um, which fell along the lines of, so we know that the amount of rock area that we have to sample from is a huge constraint on the diversity that we have in our fossil record. Is there a way that we can use this information to correct our diversity curves? Or kind of say, if this effect of the constraint of the fossil record wasn't there, what would the true diversity curve look like? And so this arose through the use of residual modeling, where we can actually kind of mathematically attempt to remove the effect of one curve on another and then see what remains within the residuals. So this was kind of first presented by Smith and McGowan. To kind of briefly go over what you're seeing here, particularly in the top left, we can see that we have an observed fossil curve in the red dots. So if we just look at the number of fossils that we have through time without any kind of corrections, how much genus diversity do we see in those? And then if we go back to how much rock area we have through time and remove that effect from that curve, how much did that curve then change? And this is what you're seeing in the blue curve. So there are particular points in time where we're seeing that the estimated diversity changes a lot when you attempt to take into account this effect of how much rock area you have to sample. So this is something that was done a lot in kind of the early 2010s, but is something that is not so popular anymore. And so there's a few reasons behind this. One of which I think is kind of largely theoretical, if you will, and it was kind of particularly led by this paper by Shannon Peters, talking about the fact that the amount of rock area that we have to sample from and the amount of marine diversity that we have can both be controlled by common signals. And so, for example, you can imagine that if we have sea level change, for example, then as sea level rises, we might expect there to be more area of shallow shelf, which enables a larger area of rocks to be preserved within that shallow shelf. But then also we have an effect where more shallow shelf provides a larger amount of habitat for marine animals and could facilitate further diversification. And so if you try to remove this effect of rock area from that of diversity, then you will inadvertently also remove some of the organic signal of the change in diversity. And this is a really big problem. And actually, Alex Dunhill in 2017 published this paper using simulations where he demonstrated that essentially exactly this, that if you attempt to remove the effect of the rock record from diversity curves, then you lose the true signal. And what you end up with doesn't really mean anything, or at least it's no closer to a true diversity curve than what you had before. So now I'm going to move on to talking about mechanistic models. So there are many different I guess, manifestations of mechanistic models that have been applied to the fossil record. But generally, these are approaches where we have some sort of model that attempts to describe evolution. This may include diversification processes, they may include sampling processes, and we use these in order to try and understand the data that we have in order to kind of extract diversity from that. So a kind of classical one that is actually incredibly classical in the ecological literature is capture, mark, recapture. It's been around for over 100 years, I believe. The concept is quite simple. So you can imagine it's based on the idea of an ecological field season where you go out to a beach, you find some snails. You might find these six snails on the left-hand side, and then what you do is you put some sort of mark on those, you put a red sticker on them, and then you come back at a later time, and then you say, I'm going to find all the snails I can again. And this time, I'm not just interested in how many snails, but I'm also interested in how many snails with a red sticker, because then I know that in this example, three of my snails are the snails that I saw last year, and then all the other snails are new snails. And now this is kind of enough information for you to glean some information about the turnover of individuals in your community, and therefore how big the population may be. And so in this case, we're kind of using capture, mark, recapture on an individual level. But in paleontology, we tend to use this same framework where each of these dots correspond to a species. And so we resample the data that we have and use this information to make an inference about the total number of species that existed. So another kind of family of models that are commonly used in paleontology, as I mentioned, kind of have these three core elements. They have some kind of birth or speciation process. They have a death or extinction process, and then they have some sort of information about sampling. So this schematic diagram that you can see from Wright et al. This is kind of broadly corresponding to the type of diagrams that you might see in RevBase for building phylogenies. But the elements that you can see here are quite common in a lot of these mechanistic models that we use. An origin time corresponding to when our clade first arose, parameters that describe speciation and extinction within the clade, some sort of information about how often we are observing the species within our clade, so a fossil sampling rate. And then here we also have an extant sampling probability. So I mentioned before, if we're using genetic information also to build phylogenies, for example, then this is where information on the sampling in that regard also comes into the model as well. So, kind of, I would say probably the most famous and probably one of the most well-used methods within this family would be pyrate. So this has been particularly developed by Daniela Silvestro from 2014. So in the top left, you can see a schematic that kind of roughly describes how pyrate works. So what you put into pyrate are fossil occurrences. And so these correspond vaguely to something like you can see in the top half of this diagram, where for a single species, you might have a series of points through time where you have samples of that species. And then you have this for however many species you want to include in your model. And this is kind of the raw data. And then the actual kind of mechanistic model of pyrate, it uses what is described here as an underlying or unknown phylogeny. So the model assumes that there is some kind of tree-shaped process, some kind of branching process that generated all of these species within the data set. And it uses a Bayesian approach to try and estimate the different parameters that describe this branching process. And so one of the things that's really nice about this approach is that it gives you a sense of uncertainty. This is something that comes hand in hand with a Bayesian approach. So in this bottom right-hand example, this is one of the earliest examples of using pyrate, where they tried to estimate changes in the diversity of angiosperms through time. And so you can see here that we have median values shown using the kind of lines in the middle. And then we also have these kind of grayed out for the right-hand side, but also in blue and green as well. These senses of uncertainty, these bounds within which we expect our true answer to lie if the assumptions that we make in our model are true. And so a lot of the people that have worked with Daniela over the last few years have done pretty amazing things in order to build on these ideas and expand them. For example, Torsten Half has done some amazing work incorporating machine learning into Pyrate. This is an example in the bottom right here, looking at the evolution of elephants, and particularly their coevolution with humans and how likely it is that the presence of humans actually corresponded to a higher extinction rate in the clade. So the kind of idea here is that the kind of framework of Pyrate is very similar, but some of the tuning elements, the speed, these sorts of things are improved using machine learning. And then I've also been involved in the development of Deep Dive and Deep Dive R, which are these approaches which estimate like diversity, so species numbers through time, whereas Pyrate is mostly focused on estimating rates, speciation and extinction rates in particular. So to give another example from my work, so I kind of showed this example from phylogenetics before. One approach that uses phylogenetics, we would term something called phylodynamics. This is essentially an approach where we use the shape of a phylogeny in order to infer things about the population which it aims to describe. So within our kind of paleontological ways of thinking, you can think that you can build a phylogeny of a fossil clade, for example, and then phylogenetics will tell you, based on this phylogeny, estimates of speciation and extinction through time, the total number of species, or the total number of branches in the true tree, things like this. And so this is kind of roughly described in the top left. We can see we have our schematic phylogeny at the top. And then if we have a series of discrete time bins, we might be able to bin this phylogeny into those time intervals and say, right, within this interval, we can count that we have one speciation event and one extinction event. But then these approaches are typically Bayesian, and we use that framework in order to say, well, what about all the things that we haven't sampled? And then ultimately, this gives you an estimate of, for example, at the bottom here, speciation and extinction rates through time. And so this is an approach that I used in my own work during my postdoc at ETH Zurich. So in the bottom right, you can see we looked at diversification rates in dinosaurs, particularly being interested in the lead up to their demise at the end Cretaceous, and trying to infer whether what we think we know is because of the fossil record or true dinosaur diversity. And essentially, we found that it's due to the models, it's due to the assumptions that we make in our models, and that really we need to work on making these as realistic as possible if we want to be able to get a reliable answer to whether dinosaurs were already in decline before this mass extinction event. So just a kind of side point to briefly touch on to wrap up this kind of part of the talk. I also want to mention most of the methods that I've talked about have a real focus on biases through time. I think particularly because this is what most people are interested in calculating, changes in global diversity through time. And actually there's been less of a focus on how we can address differences in sampling and diversity across space and how we can handle geographic bias. I guess Daniela Silvestro, particularly in Deep Dive, has tried to kind of take this into account by creating some kind of spatial compartmentalization within the model. But essentially, this is really difficult to do. One avenue that I think is really interesting is there's this field of methods called occupancy models. Again, these are kind of broadly drawn from modern ecology, where a model is used to try and parameterize this difference between the number of fossils that we have and different elements of sampling bias and compare these to kind of true diversity. But at the moment, these models mostly give you information about how good the sampling is, rather than actually being able to tell you anything about the true diversity. But I think this is a really exciting avenue, and I'd be interested to see whether these two approaches can kind of be brought together better in the future. So, to kind of sum up this part of the talk, the kind of pros and cons, it seems that um, based on what I've seen in the literature, simpler methods are much easier to implement, and ultimately they require less computational resources. But I think most people would also like to have a sense of whether this compromise also leads to a compromise on accuracy. Um, Subsampling and extrapolation methods are popular, but while subsampling methods can be criticized for discarding precious data, extrapolation methods may instead be amplifying false signals. There's this kind of outstanding question as to whether phylogenetic approaches perform better than occurrence-based ones, even though it takes a lot of time and effort and expertise to build a phylogeny. Is it actually kind of worth investing in that, and does it lead to better estimates? And then also there's this sense that the method performance and popularity may vary between clades. So particularly between people that work on marine invertebrates, where they might have a lot of data, and people who work on terrestrial clades, where you might have much less. Does that mean that there are different methods that would work better for these two different groups of people? And what does that mean for making fair comparisons between the terrestrial and marine realms? So I've talked for a long time already, so I'm going to go through this pretty quickly. But we also use this case study of Permian Triassic brachiopods, where we took data from the paleobiology database, and tried to have a look at whether these different metrics would give us kind of a different picture of what the end Permian mass extinction event looked like. So you can kind of see a brief comparison. At the top, we have this kind of raw count. So if we do absolutely no kind of morphing of the diversity that we have, we just look at the occurrences and count the number of species and genera that we have in each of those time bins. What do we get? And you can see this at the top. So we have these kind of an increase in diversity through the first half of the Permian. And then we kind of have these very dynamic changes, perhaps an overall net loss towards the Permian Triassic boundary. But then you see this really big drop off when we move into the Indian across the mass extinction event. And these levels remain quite low into the early Triassic. But then when we range through this data, we again see this increase in diversity over the early Permian. But then instead, actually, we see this kind of long decline. We see the highest diversity in the Capitanian, and then a gradual decline through the Wuxi-Pinyin and Changxingian into the Induan. And actually, if we were interested in the end Permian mass extinction event as a kind of instantaneous loss of diversity, this range 3 count actually gives us a really different picture. And so to give you a couple more examples, we also rarefied based on geography. We used squares, which is a metric that John Alroy proposed. We used the coverage-based estimate, and we also used the residual modeling approach. And these will give us kind of vaguely different curves. But yeah, I just kind of wanted to highlight that particularly, these are two examples where we have squares diversity at the top and these coverage-based estimates at the bottom, where some of these methods really accentuate this fall in diversity at the end Permian, whereas others suggest that actually there was this long decline kind of between the Wordian and the early Triassic, which would give us a really different picture of diversity and potentially the drivers of this loss of diversity. So to talk about some recommendations for moving forwards, the first that we would have is that simulations have a lot of potential. A lot of the methods that I've talked about today have been proposed, but have never really been subjected to any rigorous testing on whether they work well or when or why they might work well. And although we just have the fossil record, and it's really hard for us to then have a true signal in order to make that comparison with, this is the beauty of simulated data. We can simulate a diversity curve, subject it to sampling, and then use that relationship between these two things to investigate how our methods are working. The next thing is to have a clear and testable hypothesis. So, in comparison to our kind of controversial title in the preprint, will we ever truly know whether our estimates of diversity are correct? No, we won't. But we can have much more certainty about the answer to certain questions than others. So it's much easier for us to address hypotheses such as during which of these two geological stages was diversity higher than it is to say what did the diversity curve look like. So in short, we can think about the questions that we're asking and do it in a way that is much more reliable. The third thing is to embrace uncertainty. So, in my opinion, methods that generate an estimate of uncertainty are those which really we should be prioritizing and preferring. Also, because not just in a kind of philosophical sense that they're saying this is the uncertainty and we can consider that envelope. But it also means that we can make interpretations using that confidence interval and allows us to go beyond just these kind of medians or means that we would get from a metric that doesn't give us an uncertainty estimate. And then the last thing is that there is always more that we can do to improve the quality and quantity of our data. So more fossils are always being discovered. Many are missing from global databases. This is something that I spend a lot of my time doing. So if you would be keen to add some fossils to the paleobiology database, I would love to help you with that. Data equity is something that is becoming increasingly recognized, kind of who in the world has access to data, who in the world contributes to these databases, and how can we make that fair and advantageous for everyone. There are also things like text mining, for example, where we might be able to improve databases using automation. And then we also need improved integration between modern data and that from the fossil record. So this is a kind of brief summary of those 4 points that I just made. If you would like to take a look at the preprint, here is the QR code again. And otherwise, please do get in contact with me, either ask a question now or here is my email address, and you can also reach out on Blue Sky. So thank you very much for listening. And yes, I'd love to hear your questions.
Thank you, Bethany. This was a very informative talk. I would like to remind our audiences that if you have any questions, now is the time to raise your hand or directly put it in the chat, or you can send your question to our questions at PaleoPerks host. With that, we already have a few questions in our. Uh, so we will begin with the first question, uh, which is asking, have you looked into methodologies to estimate diversity and rates of changes that are used by biologists to look at modern species as well, aside from the capture, mark, recapture? How do these compare?
That's a really good question. So for this review, we just focused on the paleontological literature. We didn't actually attempt to survey the modern ecology literature, but absolutely there is a really big crossover. As I mentioned with capture, mark, recapture, a lot of the kind of concepts are very transferable, and I think there's a lot of potential for more exchange. I think. Is it likely that a method exists out there that no paleontologist has heard of or used yet, and yet is also somehow a magical solution to all of our problems? I think that's quite unlikely. But I do think that, yeah, more exchange between people who are interested in quantifying diversity between things that are living and things that are long dead would be really advantageous on both sides. So I'd love to see that in future.
Thank you. So we have a hand raised by Jim, and I'm trying to see how to unmute. Um, okay, Jim, you should be able to ask your question.
Okay, I'm Jim Ogg. I work with timescales and been working with various people around the world and commissioned geological map of the world. And one thing that's been interesting as we put together databases is how much the assumed ages for geological formations have changed with time. And this, of course, is going to influence your record of when does this fossil happen? Because, for example, the Tatarian stage in Russia, people assume that was the late Permian, but now they put it into the middle Permian, which means all those ages which were assigned to Tatarian have to shift. How to adjust that easily with things like the paleobiology database and other sources?
Yeah, this is a really great question and something that I don't have a kind of easy solution to. I think there's a couple of different elements there. So one is that it's kind of dependent on the way that we record data. So, for example, when most people download information from the paleobiology database, they are told that there was a fossil collected from this place, and there is an assumption that it belongs to this particular geological interval. And so, in some cases, that is the best that we have, and that the dating is based completely on biostratigraphy, in which case, in theory, the kind of moving of the ages of these boundaries shouldn't matter because it's still kind of conceptually cohesive. As long as that fossil stays within whenever we think that that interval was, that's the best that we can go on in terms of how old it is. But then there are other examples where things like radiometric dating are used, for example, where you can imagine a scenario where you have a really precise date for a fossil, and then that means that as a boundary is moved, this actually changes the time bin within within which that fossil lies, and how well our databases are set up to account for things like this. I think the answer is not very well. So I think personally, I think that biostratigraphy in itself is like quite a big barrier for us getting accurate estimates of diversity through time because there's so much kind of circular logic in the way that we date things and therefore how old we think they are, and which communities belong together. Um, yeah, I'm not sure that I have a kind of good idea for a solution to this problem, but I really do agree that it is something that can create a lot of noise and issues within the data, definitely.
Thank you, Jim, for your question and Bethany for your answer. We have a question from Sarah Saxton, who is asking, who has first said, great talk. And then she is asking, um, are there any good methods for estimating how intrinsic traits like ecology, example, phos- fossorial versus semi-aquatic, benthic versus pelagic, habitat preferences, and geographic range size might affect the probability of given species fossilizing or the relative abundance of fossils from one species versus another?
That's an interesting question. My gut feeling is no. Unfortunately, there aren't kind of good methods for estimating these things yet. I think some data does exist in which the ecology of certain fossils is detailed. But we end up in this scenario where we have fossils and we know their ecology, and because we have them, we know that we found them, but we don't have information on where we haven't found them, and therefore what the ecology of those fossils that we don't have is. And really, what this requires is quite a big kind of predictive model, if you will. I kind of mentioned occupancy modeling very briefly. So, this is a framework where you can use things like particular attributes of certain fossils or certain classes of fossils and what this means in terms of whether they are more or less likely to be preserved. But as I say, this this field kind of requires quite a big modeling framework because you need to infer a lot about the information that you don't have. And this means that you need a lot of data actually in order to be able to feed these models well. They're very data hungry. And so if you work in quite a small system where you don't have a lot of data, yeah, it's very difficult to use these methods. So yeah, unfortunately, I don't have a good solution for that one, but I'd be really interested to see one in the future.
Thank you, that for the honest answer. We have another question from Livia, who is saying, "Great talk. Do you think these methods for quantifying biodiversity changes can be useful for looking at tipping points in the microfossil record, or the issue of volume of data makes this difficult?"
That's a great question. So, I think I kind of illustrated very quickly in my case study. Maybe I can go back a few slides. Yeah, this idea that when we looked at the brachiopods across the Permian and Triassic, depending on which method you used, either you would see this really steep decline at the end Permian mass extinction event, or this gradual decline that actually happened kind of over most of the end of the Permian. And so, yeah, when it comes to this question of tipping points, this exactly is really, really important because if you looked at that kind of upper graph, you would say, you know, this is a tipping point. There's some sort of threshold that we've crossed. We've seen this massive crash in diversity, and that kind of suggests that something very severe has happened, which has led to all of this extinction happening in a very short period of time. Whereas if you looked at this bottom graph, you might instead say, well, does this correspond to the crossing of a tipping point? Probably not. You know, we're seeing this decline over tens of millions of years. Maybe this is a driver which has happened more slowly over a more prolonged period. And so, yeah, I think it depends on specifically the system that you're interested in. I guess whether, you know, a tipping point might be, I suppose in this context, a climate tipping point, for example. But yeah, I think it probably does have really big implications. I would say, so I believe the question was specifically about microfossils. I mean, working with microfossils, you already have the benefit of a much more abundant fossil record than some of the rest of us work on. And I think that's always going to be in your favor. It gives you more choice of methods, and most of these methods are probably going to perform better when you have more data. Um, so I think maybe that means that you can lead toward things that are more computationally intensive, but also it means that you can compare more different methods. I guess you're going to have more conviction about whether a tipping point has crossed if you use four different methods and they all say that you have, in comparison with if you only use one.
Thank you for the thoughtful answer. And we have this anonymous question, which is again saying, "Great talk, very informative. I'm inclined to understand the association of science and society and was wondering if you could reflect on what the narrative or story is generated from the study of deep time biodiversity." Then there is a second question: what are your motivations for finding new ways to correctly quantify deep time biodiversity?
That's a really interesting question. So I guess quite a lot of these methods um, were initially presented in papers that actually don't have a lot of authors. So there may be kind of solo author papers or kind of two people working together, which suggests that they often arise by just isolation almost, like somebody just sitting down and thinking really hard about a question, which I think there can be a beauty to, but also, you know, at least in many other fields of science, we're kind of moving in the opposite direction to that. A lot of amazing science is done when these huge collaborative projects come together and lots of people give input. So I think in terms of developing biodiversity metrics, certainly the past very much seems to look like, um, yeah, kind of individualistic thinking, someone having an idea, thinking about how they would implement it mathematically, and then describing a framework. I will say as well, often in a way that is not very accessible. And sometimes this is a hindrance on the wider community adopting something. If a method is presented in a paper where only 10 other people in the community can understand what the paper says, then I think that can be inhibitory to us moving forwards as a field. And I'm a big advocate, even in computational work, to trying to write things in a way that is accessible and understandable. And yeah, I think having more conversations, trying to approach this question in bigger groups, I think as well could pay dividends. But I would love to be part of that experiment. I would love to kind of get a group of people together who I thought had interesting ideas, and sit them down and see what we could come up with. I guess so. The second part of this question, right, was about my own motivations. So I guess I kind of mentioned a couple of times through the talk that I have done previous work that kind of touches on this question a lot. And so I've reflected on my own work and the work of others in this area quite a bit. I think it's just such a fundamental question. Like, understanding diversity is something that brings so many of us to this field in the first place, be it a love of dinosaurs and wanting to understand the end Cretaceous mass extinction, or, for me, I had a real love of Pokemon as a child, and so biodiversity is something that I've always enjoyed and been interested in, and I guess has kind of come back around and re-manifested itself in my science. But yeah, so just being able to say to people, if I want to compare, as I said at the start of this talk, the Cambrian and the Cretaceous, which one had more species in it? What's the trajectory over the Phanerozoic? What does this mean for how we understand evolution? I think, yeah, biodiversity is so fundamental to that, and the sooner we can get to good answers to that question, the better.
Thank you for a very poignant point you made about the accessibility of research, and also for giving us such a frank answer. So I think this has brought us to the end of our Q&A session, although there is David Saka who's asking, uh, how do I contribute to this research? Where do I start from? David, I would highly recommend joining us for the informal tea time. And thank you, Bethany, and the audience for asking uh, such good simulating questions. I hope we can continue this simulating conversation for our tea time. So, for in conclusion of our formal part of the seminar, thank you everyone for joining us. We have again dropped the link to the weekly feedback form in our chat, so that we learn more about who has attended today's seminar. And next week, September 9th, we'll be joined by Jeet Majumdar from Jogmaya Devi College in India, and he will be talking to us about paleoceanography. Uh, so, yeah, up next is tea time. So, and the question of our day is, what is your favorite deep time trivia? Like any fact or anything that you know about deep time and you would like to share. So, um, walk around, go up, walk around, can come back in 2 minutes. Bring your paleo pets, coffee, tea, anything you wish. See you in 2 minutes.